Welcome to the PubMed Daily Review for March 28 SD. Today we scanned 3,975 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5. Individualized mRNA vaccines evoke durable T-cell immunity in adjuvant TNBC by U. Sahin and O. Teresi from BioNTech Group, Mainz, Germany. Let's break down its key contributions. Triple-negative breast cancer is a particularly aggressive form of breast cancer, often associated with a high risk of relapse, even in the early stages. Traditional treatments can be challenging, and there's a critical need for innovative therapeutic strategies. This study explores the potential of individualized mRNA vaccines, designed to target unique mutations in a patient's tumor to stimulate a robust and lasting immune response against TNBC. This study involved 14 patients diagnosed with triple-negative breast cancer who had already undergone surgery and received neoadjuvant or adjuvant therapy. The researchers administered an individualized mRNA vaccine tailored to each patient's specific tumor neoantigens. They then meticulously analyzed the peripheral blood to assess the immune response, focusing on the magnitude, functionality, and persistence of T cells targeting these neoantigens over time. The results were highly encouraging. Nearly all patients mounted significant T-cell responses against the targeted neoantigens, and remarkably, these responses remained functional for several years post-vaccination. 11 out of the 14 patients stayed relapse-free for up to 6 years. While 3 patients did experience recurrence, their cases offered valuable insights into potential immune escape mechanisms, such as low MHC class I expression. This study demonstrates the exciting potential of individualized mRNA vaccines as a viable strategy for treating triple-negative breast cancer. It highlights the ability of these vaccines to generate long-lasting, functional T-cell immunity. The findings also underscore the importance of understanding and overcoming immune escape mechanisms to further enhance vaccine efficacy. Future research will focus on refining these approaches and exploring combination therapies to maximize benefits for patients. Up next, our second paper out of 5, retooling of gamma-delta T cells for cancer immunotherapy using advanced manufacturing and genetic engineering by Benjamin J.L. Lim and John Marr from University of Edinburgh Medical School. University of Edinburgh, Little France Crescent, Edinburgh, EH16 for UU UK. Let's break down its key contributions. CAR T-cell therapy has shown remarkable success, but its autologous nature makes it difficult to scale and less effective against solid tumors. This has led to significant interest in alternative cell types. Gamma-delta T cells are a promising candidate because they have natural anti-cancer properties and can be used as an off-the-shelf allogeneic therapy, potentially overcoming many of the current challenges. This paper is a review, so it doesn't involve a specific patient cohort or intervention. Instead, it synthesizes findings from numerous studies. The focus is on how advanced manufacturing and genetic engineering techniques can be applied to gamma-delta T cells to improve their potential as a cancer immunotherapy, comparing them implicitly to current alpha-beta CAR T cell therapies. The review highlights key characteristics of gamma-delta T cell subtypes. V-delta 2 cells are the main type found in the blood, while V-delta 1 cells are more prevalent in tissues. Both have shown potential for cancer immunotherapy. Crucially, innovative genetic engineering and manufacturing approaches are being developed that could significantly boost the potency of these cells for therapeutic use. In summary, gamma-delta T cells represent a compelling alternative for cancer immunotherapy, offering advantages in scalability and applicability to solid tumors. The ongoing advancements in manufacturing and genetic engineering are key to unlocking their full potential. While promising, continued clinical research is essential to validate these approaches and bring new gamma-delta T cell therapies to patients. Up next, our third paper out of 5, development of PROTACs for targeted degradation of oncogenic TRK fusions by Saurav Kumar and Behnam Nabet from Human Biology Division, Fred Hutchinson Cancer Center, Seattle, Washington, 98109, USA. Let's break down its key contributions. Oncogenic TRK fusions are key drivers in many cancers, particularly rare pediatric and adult types. While first- and second-generation TRK inhibitors have shown promise, resistance mutations frequently emerge, limiting their effectiveness. This highlights the urgent need for new therapeutic approaches that can overcome these resistance mechanisms and provide durable cancer control. To address this, researchers developed novel PROTACs, which are designed to hijack the cell's own protein degradation machinery. They specifically engineered a PROTAC named JWJ01-378 by linking the TRK inhibitor entrectinib to thalidomide, which recruits the E3 ligase cereblin. This molecule was tested in various cancer cell models, including those harboring the TPM3 TRK fusion, to assess its ability to degrade the target protein and its downstream effects. The study found that JWJ01-378 was highly effective, inducing potent degradation of the TPM3 TRK fusion at nanomolar concentrations. This degradation led to a significant suppression of downstream signaling pathways like PERK and ultimately reduced cancer cell viability. Importantly, JWJ01-378 demonstrated remarkable selectivity, avoiding degradation of key cellular proteins and showing minimal off-target effects, unlike a comparator compound. This research demonstrates that targeted protein degradation using PROTACs is a viable strategy for combating TRK fusion-driven cancers. JWJ01-378's ability to selectively degrade oncogenic fusions while sparing essential cellular components represents a significant advancement. While it did not degrade certain resistance mutants, this work expands the therapeutic toolbox and warrants further clinical investigation for patients with TRK fusion-positive cancers. Up next, our fourth paper out of 5. High-accuracy stool biomarkers of precancerous colorectal cancer identified using a 2,000-plex immunoproteomic screen by Kamala Vanarsa and Chandra Mohan for Department of Biomedical Engineering, University of Houston, Houston, Texas, USA. Let's break down its key contributions. Colorectal cancer is a major cause of cancer-related deaths worldwide. Early detection is key to improving survival rates, but current screening methods aren't always effective or accessible. This study aimed to discover new, accurate biomarkers in stool that could help us catch precancerous changes before they become full-blown cancer. Researchers used a high-throughput proteomic screen to analyze over 2,000 proteins in stool samples. They compared samples from patients with colorectal cancer, those with advanced adenomas, and healthy individuals. Promising candidates were then validated using ELISA across multiple independent cohorts and ethnicities to ensure reliability. The study found that a panel of stool proteins, including MMP-8, MMP-9, hemoglobin, PGRPS, and fibrinogen, were highly effective in distinguishing colorectal cancer from healthy controls, with AUCs ranging from 0.91 to 0.95. Even more impressively, these biomarkers could also differentiate advanced adenomas from healthy controls, with stool fibrinogen alone achieving an AUC of 0.86. This indicates a strong potential for early detection. This research presents a promising step towards developing a non-invasive stool-based test for colorectal cancer screening. The identified biomarkers demonstrated excellent accuracy, potentially improving early detection rates. While these results are exciting, further clinical validation is essential before these markers can be implemented in standard patient care. Up next, our fifth paper out of 5. Tabular LLMs for Interpretable Few-Shot Alzheimer's Disease Prediction with Multimodal Biomedical Data by Sophie Kearney and Li Shun. Let's break down its key contributions. Diagnosing Alzheimer's disease accurately is a major challenge in medicine. Traditional methods often struggle with the complex and incomplete biomarker data we collect. While deep learning shows promise, it can falter with small datasets. This is where the power of large language models, or LLMs, comes into play, offering new possibilities for prediction and understanding. To tackle this, the researchers developed TAP-GPT, a specialized LLM for tabular data. They fine-tuned this model using prompts derived from tabular biomedical data, including biomarkers, MRI, and PET scans. The goal was to see if it could accurately predict Alzheimer's, especially in a few-shot learning scenario where data is scarce. They tested it across multiple datasets to ensure robustness. The results were quite promising. TAP-GPT significantly outperformed its base models and traditional machine learning approaches in predicting Alzheimer's, particularly when only a few examples were available. Importantly, it handled missing data gracefully without needing imputation, a common hurdle. Furthermore, the model provided structured reasoning that aligned with our current understanding of Alzheimer's biology, adding a layer of interpretability. So, what does this mean for the future? This study highlights the potential of specialized LLMs for tackling complex clinical prediction tasks, especially with limited data. TabGPT's interpretability and robustness to missing data are key advantages for real-world application. This research paves the way for more advanced, AI-driven decision support systems that could revolutionize how we diagnose and manage diseases like Alzheimer's.